Hello, everyone. This is Dr. Susan Marra with Dr. Marra's Medical Minutes. And today we're going to talk about the differences between the tests that we use to identify the tick-borne illness infections. So the first one that we use is the Western blot or Immunoblot, and that is a measure of antibodies against an organism and those antibodies are produced by your body. They're part of the B cell wing of the immune system, and B cells are made in the bone marrow. And B cells are really important for making antibodies. And antibodies are molecules that actually bind to the antigen or the microorganism and help to neutralize its effects. And so we see IgM, IgG, IgA, and IgE. So IgM and IgG are what in general are used to identify the presence of a microorganism. IgA antibodies indicate an infection in the mucosa of your body. And IgE antibodies are generally antibodies made to allergens, food allergens, environmental allergens, things like that. So obviously antibodies are a really important part of the immune system. And that is what we measure on a Western blot or an Immunoblot. The other test that we use is called PCR. And PCR stands for polymerase chain reaction, and that's a test that actually measures the DNA of the organism. So if you have a positive PCR test, that means that they found the DNA of the organism in your body. Now, it doesn't tell you if the organism is alive or not. It's telling you about the DNA of the organism. So some people say that, a positive PCR doesn't necessarily mean that there's a viable living organism, because it could be a piece of dead organism that remains in the body after significant killing. So there's fragments of DNA from the organisms. So that's another problem. But, what you can pretty much do is you can tell by the patient's symptoms whether or not there's an organism present. A seasoned practitioner would be able to tell that. And then another test that we use in particular to identify Bartonella and Babesia, it's called the FISH test, flourescent insitu hybridization. And this is a particular test that actually documents on a slide the presence of Babesia or Bartonella. And it's a very useful, very reliable test. And I like to use it to document Babesia. I think it's the most sensitive test for Babesia. But I also use antibodies as well, because antibody, specifically the Immunoblot, is going to tell you the different species of Lyme, whether it's European or US. It's going to tell you the different species of relapsing fever. Is it Borrelia miyamotoi, Borrelia hermsii, Borrelia terracota? What is it? And then the different species of Bartonella, Bartonella henselae, Bartonella elizabethae, Bartonella quintana, and so on and so forth. And then Babesia, the different species of Babesia, Babesia duncani and Babesia microtii. Those are the species that IGeneX can pick up. So, when you go to a Lyme literate physician and ask them to test you for tick-borne illness, they're likely going to choose one or all of those tests to help document the presence of an infection. And the reason it's important is that, for example, in the state of Washington, you may not treat Lyme disease with antibiotics unless you have positive tests. With the exception, if a bullseye rash was present and you have a picture of that on file, then that is pathognomonic for the presence of Lyme disease. So that is a very good thing to capture. So if you ever have a tick bite and you have a bullseye rash, take a picture of it and take it to your doctor. It's really important and it's really, really helpful. So I use all of these tests, and but I would say that I rely heavily on the Immunoblot and the fish test. I do PCR when I I feel like I can, because PCR is one of the hardest ones to get a positive test for. So I hope this information you have found useful. I hope you have a wonderful day. Take care.